Assalamualaikum student. Today we are going to learn 10th standard chapter 1 Heredity and Evolution. In this chapter we are going to discuss how heredity is responsible for evolution in a living organisms. In our previous standards we have already discussed about the cell and the structure of a cell. Inside the cell we have seen there is a nucleus, and inside the nucleus there are chromosomes which are present. These chromosomes are coiled with a DNA molecule. Inside a DNA molecule there is a specific coding which is responsible for our characteristics, inherited characteristics, and that specific length of a DNA is known as genes. Hereditary is a characteristics transfer of a biological characteristics from one generation to another generation via genes. Hereditary is a process in which organisms acquire characteristics from their parents. These characteristics are called traits. Every individual is unique because they have a unique sets of traits. The traits which are transmitted by the parents to its offspring during the process of reproduction. And this inheritance is determined by a certain rule of heredity. Inherited traits are coded in a DNA. In our first part of a chapter, we are going to see how this DNA coding helps to synthesize a different protein in a living organism. DNA participates in the functioning of the cell with the help of RNA. In our 9th standard, we have already discussed about different types of RNA and their structure. We discussed about messenger RNA, ribosomal RNA and transfer RNA. With the help of RNA, genes present in the form of DNA participate in functioning of cell and thereby control structure and functioning of the body. Appropriate protein required for our body is synthesized with the help of DNA. Information which is stored in DNA will help to produce protein molecule with the help of RNA. Information about protein synthesis is stored in DNA. As per the requirement, protein molecules are produced, and this synthesis of protein is through RNA molecule known as a central dogma process. First steps in a protein synthesis is transcription process. In a transcription process, mRNA will produce with the help of a coding on a DNA. Here the DNA molecule will open up and there are two strands of a DNA molecule. In mRNA production, only one strand of a DNA molecule will participate. Inside a nucleus, along with DNA molecule, there are different nucleotides which are present. As soon as this DNA molecule strands will open up, these nucleotides will get attracted toward the DNA strand. Here there is a thymine. So thymine, the complementary for thymine is adenine. So adenine nucleotide will come and attach over here. Complementary nucleotide will attach with nucleotide. Guanine will attach with cytosine. Then adenine will attach with thymine. But as you know, in mRNA there is no thymine. Instead of thymine we have a uracil molecule. So here to this adenine, uracil molecule will attract. Here the different nucleotides attract, and after there is a formation of mRNA molecule, and afterward again this DNA molecule will coiled up. Here the mRNA is produced in this process. So here the mRNA is produced as per the sequence of nucleotide on DNA. Only one strand of a DNA is used in this process. The sequence of nucleotide on mRNA being produced is always complementary to DNA strand, and beside this there is a uracil instead of a thymine in a DNA molecule. This whole process is known as a transcription process. mRNA produced in this transcription process inside the nucleus will first come outside the nucleus in the cytoplasm. It brings the coded message of a DNA. The message contains the codes for different amino acid. The codes of each amino acid consist of a 3 nucleotide and it is called a triplet codon. Here the one of a amino acid lysine is carried through this 3 triplet codon. Here you can see methionine is another amino acid which is carried by another 3 nucleotides. So that is known as the triplet codon. This triplet codon helps in a formation of a protein molecules. Each mRNA is made up of thousands of a triplet codon. Here there are different triplet codons which are present. As per the message on mRNA, amino acid is supplied by tRNA. First of all, when the mRNA comes in a cytoplasm, ribosomal RNA attached with this, and the amino acid carried by tRNA molecules. tRNA molecule contain different amino acid. So this tRNA will bring amino acid as per the codes over here on mRNA. Let us consider this is one of a code on mRNA, A.A.K, adenine. So for adenine, which one is the anticodon? Anticodon will be a uracil, UUU. So here in this process, each mRNA is made up of thousands of a triplet codon. As per the message on mRNA, amino acid is supplied by tRNA. For this purpose, tRNA has anticodon, having a complementary sequence to the codes on a mRNA. This process is known as a transcription process. As per next sequence over here on mRNA, another tRNA will bring amino acid. Here there is a guanine, adenine and uracil. So for guanine, anticodon will be complementary codon, nucleotide will be a cytosine. For adenine it will be uracil, and for uracil it will be adenine. So the next tRNA molecule will come and attached over here. Now when these two tRNA attached here and these two amino acids come close to each other, there will be a formation of a peptide bond. So here there is a peptide bond which forms between these two amino acids. As soon as there is a formation of a peptide bond over here, this tRNA will leave. Now then this ribosomal RNA will move ahead and the next tRNA will come and attached over here. Here the coding is UUC. So for uracil there is a adenine complementary, again adenine, and then for cytosine there is a complementary nucleotide guanine. So this nucleotide will come, tRNA will come and attached over here. Okay, now here these amino acid, when it comes closer, what will happen? There is a formation of a peptide bond again, and in this way this process will continue. The amino acids brought by tRNA are bonded together by a peptide bond with the help of RNA. During this process, ribosomes keeps on moving from one end to another end of mRNA by the distance of one triplet codon. This whole process is known as a translocation process. This process will continue till the required protein will synthesize. So in our today's lecture, we have discussed about protein synthesis in a living organism. First steps in a protein synthesis of a living organism is a transcription process. In this process, mRNA is produced inside a nucleus with the help of coding on a DNA molecule. mRNA which produced in this process has anticodon, complementary coding to the DNA molecule. This mRNA which produced in a nucleus come outside the nucleus in a cytoplasm. Then the ribosomal RNA will attach to this mRNA and tRNA then supply a required amino acid as per the sequence on mRNA molecule. Then there is a formation of a peptide bond between these different amino acids, and finally these amino acids then rolled into a polypeptide ball, and then it forms a protein molecule. Thank you, everyone.